Hey guys, looks like the stream is working. Let me know in the live chat if there's something wrong with the audio or video. As far as I can tell, it's working on my side. So I decided to quickly come on live again because I was looking at another pond water sample. I let this one sit under a light for a few days, and everything tends to kind of precipitate to the bottom, which makes it easier to pick up with a little pipette. So there's a lot of heavier animals in this one. I believe the large protist in the center or near the center of the screen is a colpidium, which is a ciliate. You can see it has cilia all over its body, which it uses for locomotion as well as to move food towards its mouth or whatever it happens to use for a mouth, the opening to its digestive system. It's a single-celled organism, which makes it quite huge for a single cell, considering that the smaller kind of worm-like creatures around it are rotifers, which are multicellular organisms. So we have a large blobbish single cell, which is many times the volume of these multicellular animals. It's really kind of bizarre, the scale of some of these little creatures. If you look closely, you can see as he slides in between the algae and the glass, you can see all of his insides kind of squish around. They're not fixed in place like the organs in our body. They can kind of move around on their own internally. So this was one of the interesting creatures that I found and wanted to show today, as well as some Stentor. And I've seen a Stentor once before in the microscope, but it was only one on its own. In this particular sample, there's quite a few, and we're going to look for them in a moment. There's a lot of roundworms in this as well. You can see that long, skinny worm moving side to side. It's also covered with cilia that it uses to move around. There are also a lot of small crustaceans in this sample you'll see floating around. They're quite lively. So stentors are actually some of the largest single-celled organisms. They're kind of trumpet-shaped. They anchor themselves to some substrate and kind of wave around in the water to try and capture some prey. I have no idea what this is, but it looks like it may be either budding, a single organism splitting, or it's going through some kind of conjugation where it can share genetic material. Some single cell organisms will share genetic material, others will just bud and produce clones of themselves. Oh, it seems like one of them has been punctured, so this may not be a positive interaction for one of these organisms. Alright, so I'm still trying to find the stentors that I had seen earlier. Oh, this is off the edge of the slide. Here is the extreme limit. Lots of rotifers. Oh, that looks like a paramecium. kind of wiggling side to side next to the rotifer. Paramecium has kind of a partially spiral shaped groove on its body, I guess you'd call it, that it uses as an opening for its digestive process. So again, using its cilia, it will pull water and material into its digestive system. Oh, there's a Stentor right there. So once he stretches out again, I'm gonna move the scale over so we can see how big he really is. Actually, let's go up one magnification. I haven't been able to identify the little tiny ones yet, mostly because they're too quick. If I see one sitting still, we can try and get a closer look. But here you can see the water around the head end of the Stentor kind of circulating, and that's how it would filter feed material out of the water around it. We may have caught something. Yeah, those little ones are moving so fast I can't even tell whether they're using cilia or a large flagellum to kind of whip their way through the water. Some protists will have their tail on the back and lets them wiggle like a fish, and some of them will have the tail on the front, which lets them kind of corkscrew their way through the water. At such small scales, the viscosity of water is very different. So for us, water seems to pour quite freely, but for these tiny animals, water is actually quite viscous. It's kind of like swimming through molasses. So things like holding your hand in front of you and wiggling it side to side could pull you through a viscous liquid, but doesn't really help you with water. That's why some of these small creatures have very strange ways of moving compared to how we're used to thinking of swimming through water. I think I can see on those small creatures a flagellum on the front of the body. Which means they can pull themselves forward through the water. There's a couple of them next to this spirogyra. That's the stripe going across the screen. I'm going to try and go to bright field and get a better look. Oh, the Stentor is just retracted. I think I bumped the microscope and he felt the vibration. So it looks like those small ones may still be ciliates. I think I can see little rows of dots. I have a little identification open this time, so hopefully I can give you some names to go with these guys. Oh, they've all scattered. Some of these are actually sensitive to light, so if I look at them in dark field, they don't really mind, but when I switch to light field, they all kind of scatter. So let's zoom out again and see if I can find more of those Stentor, because there was quite a few of them together. The really wispy hairs, I'm not sure whether it's an algae or it might actually be a bacteria. It's so small I can't really tell, but this microscope is just barely able to see bacteria. You might see little corkscrew shaped hairs, kind of pigtail shaped hairs that will screw themselves through the water, and these are spiral bacteria. They're about the same width as the little hairs that seem to be in the background. The other thing that makes me think it might be bacteria is this sample has been in the jar for a day or two, undisturbed, and when you have a closed jar of water filled with pond muck, it just tends to, grow bacteria. Here's one of the larger Stentors that I saw. So this guy is over a millimeter long and is only a single cell. You can kind of see along his body there's a shimmer. That's more smaller cilia that coats the side of his body. You can also clearly see the mouth opening and the way that the cilia kind of rotate towards it. Super AWAC. This is an eBay special microscope. You'll see them sold as pathological microscope or veterinary microscope. The quality is pretty good for what you pay for. There's certainly a lot better microscopes available, but if you're going to use this just as a hobby scope, it's more than adequate. The Stentor is stretched out now. He's probably about 2 to 2.5 millimeters long, which is unusually large for what you'd think of for a single cell. Yeah, the long trumpet-shaped one is a Stentor, and it's actively trying to filter water into its mouth parts so that it can get a meal. You can kind of see, I'm not sure if you can, but he's got a lot of stuff spiraling around his mouth opening, and if we get a close look, we might actually see him capture something. Brain for nothing. In order to get the scale, I actually have a loop. It looks like a jeweler's loop, but it's actually for printing. And on the glass cover, there's a very, very tiny etched scale, or printed scale rather. And I've taken static pictures from the video and then created the scale that we see at the bottom. I'm not exactly sure how they hold on. I think they produce some kind of adhesive on their foot. There's a lot of... Oh, see, he's let go. He's become mobile. There's a lot of filter feeding creatures. I think there's a Vorticella kind of in the middle now. That looks like a Vorticella, and they use almost the same technique. They have a tether that they anchor onto the substrate, and then they stretch. Or some of these Stentor and rotifers I was looking at earlier before I set up the stream. Um, this is probably one of the most active water samples I've seen. I've never really seen so many creatures all in one spot moving around, so I wanted to quickly come on and show you guys. So here's one that's attached himself. We're going to leave the mobile one and we're going to look at this one for a minute. This one's attached himself right to the slide. So I'm going to try and zoom in and we can take a look at that point of attachment. But there appears to be a rotifer in the way. So it looks like he has just a kind of a sticky foot that they can deploy and stick onto a substrate. I don't see any kind of like on the rotifer, you can see they have kind of toes. It's kind of shaped like a V at the end of the body that they can use to kind of stick and inchworm their way across a surface. Yeah, it's amazing how uncooperative some of these little organisms can be, considering they have bodies less than a millimeter wide. They can still block an entire shot when you're using a microscope. Well, you can see a little bit better here the mouth opening on the Stentor. He was kind of looking straight up at us, and it made it visible. You can also see all the organelles inside his body mixed in with anything he's captured and currently digesting. We also get a really good look at the only hard parts in the rotifer's body, those teeth that are more or less in the area where your neck would be. So it captures food particles out of the water and then grinds them up using those little teeth before passing them to its digestive system. Oh, there's the edge of the slide. We're in the corner. Oh, here's one of those little crustaceans. So this one has a single eye spot, that red spot in the middle of the body, as well as a pair of arms that it uses to kind of swim through the water. If I can, I'll try to keep him in frame and find a name. There's quite a few of them in this sample. They're usually hard to catch because they like to swim around, and it's like trying to grab 0.2 of a millimeter of an animal with tweezers and not squeeze them. Oh, if you'd want to see a really good video that quickly covers a lot of different types of these little creatures, look up the diversity of protists. It's on YouTube. It's an older educational video, but it goes over the names of almost all of the things I've seen in pond water in my area. There's a couple things I haven't seen, but for the most part, the diversity of protists video covers everything you'll probably run into in a pond water sample. So also in this sample, I'll try to find, there is a larva for some kind of fly. I'm not sure what it is, but it's actually, compared to what we're looking at here, it's enormous because it's on the multi-millimeter length scale. It makes it a giant compared to these. Alright, so the best name I can give you for this little crustacean is a copepod, which means oars for feet, and given the way he's swimming, that kind of makes sense. Nevin, this sample came from across the street. There's a stormwater pond. They put those in a lot of residential areas here because they build up so quickly with houses that the stormwater system can't really handle it. So they will put a stormwater pond every now and again, and basically all the runoff from streets and all the runoff that comes from properties onto the street all collects in these stormwater ponds, where it can evaporate or slowly soak back into the water table. So although it's going to be some of the least chlorinated water, all the chlorine is going to have a chance to kind of work its way out, as far as from watering lawns and whatnot. It is going to have a lot of contaminants from the streets. There'll be a lot of oil contaminants, but it doesn't really seem to bother many of these guys. So I'm looking for that large worm right now. Actually, there's a large roundworm crossing almost the whole screen, but that wasn't what I was looking for. This guy stays the same his whole life. This is what it's going to be. It's just going to keep getting bigger. What I'm looking for is a fly nymph. It could be, or a fly larvae, sorry. It could be a mosquito larvae or some other type of fly. But he moves extremely quickly because he's really big, probably 5 millimeters long. So I'm going to quickly scan back and forth here to see if I can run into him. Oh, here's some Vorticella. Actually, let's stop and take a look at the Vorticella. They're always really cool. There we go. There's a really good look at a Vorticella and its feeding behavior. You can see it waving its cilia and churning up a little vortex of water around its mouth opening. So by sitting there and just pulling water past itself, it can filter feed. As for how I attach the microscope to the or camera to the microscope, I'm going to move the webcam here for a second. So I've got a camcorder. It's built around a mirrorless camcorder, so it's one of the Sony VG lines. There's a length of plumbing pipe, and that's actually clamped right in place of the microscope head. And then down here is the subject. So the image is actually being projected from the objective lens directly onto the sensor inside the camera. So there's no extra optics, and that's what really helps get these clear images. I might actually have to shorten this tube a little bit. The reason I made it so long is the farther away you get, the more the image is magnified. As it widens, the image gets more magnified, but unfortunately it limits my focus. So if I have very deep samples that have a lot of vegetable material in them, I can't always focus right to the bottom, right to the surface of the slide. But yeah, all that's on here is a piece of pipe and an aluminum can and a hose clamp that actually clamps the camera onto the end of the pipe so that it doesn't fall off. Let's see, here's the other Vorticella, and you can kind of see the rotating motion of the cilia. So it moves water downwards towards the body, but also in a circle around it, trying to move it towards the mouth opening. I'm still not sure what the really quick ones are yet. Oh, there we go. So the Vorticella was disturbed enough that he coiled up his little tether. You can see it uncoiling now. I measured that with my old high-speed camera to be a ridiculous amount of acceleration, like on the hundreds of G's being experienced by this tiny little animal. I believe the little hairs, they are either a very tiny algae or they may actually be bacteria. This water sample is actually sat for a little bit of time, and that kind of promotes the growth of bacteria. Oh, here's something else interesting. You can see in the middle it's 4 little dots, and you might be able to see 4 little flagellum kind of waving around off to the sides like an octopus. I'll try and see if I can get a name for that. Yeah, I always find it bizarre as well how such tiny creatures can be mechanically so complicated. You'd think that the like it would need a full nervous system with lots and lots of nerves in order to control these little complicated motions. Oh, I'm. Did I miss something? Oh, here's more. So it looks like these little clusters are colonies, which might make them gonium. So gonium will live in 4, 6, 8, 16, or 32 cell colonies. Interesting, powers of 2. It might give you a hint as to how they reproduce. Each cell has 2 flagella and an eye spot, as well as a chloroplast, which means it also can take its energy from the sun, or it takes its energy entirely from the sun. So you could think of this like a walking houseplant. It can move itself in order to get more exposure to sunlight and better survive. I've seen other small organisms like this in the ocean. I took some video a couple years ago of this green scum that was just kind of collecting in footprints on the beach as they filled back in with seawater. And when I looked at them under the microscope, they were individual cells that had a single flagella, and they could steer themselves towards the light, because when my sample started it was uniformly green, but when I finished it was a green spot about the diameter of the light on the microscope. So these little sunlight powered creatures had all motored themselves towards the light that illuminated the sample. It's very interesting, not thinking of, or I don't normally think of sunlight powered life as being very mobile. I usually think of trees or moss. So we can do one more pass and see if I can find that larva, that fly larva, and if not, I think I'm going to call it a day. So if you have any more questions, let me know on the side. After I'm done, I'm going to pick an interesting place, and I'll just let the camera run for a little more if you guys are interested. So here's another a roundworm. I've actually just finished ordering all the parts from AliExpress and eBay to try and automate my microscope setup. I want to try and make it controllable from a joystick so that the movement is a little more precise. Oh, here we can see a number of roundworms feeding on something. It looks like it may have been a Daphnia, but you can see the, I guess you call it the throat of these little worms as they're pulling material out from inside the Daphnia's body. Nevin, I've had some samples sit in a jar. I use just mason jars, in a sealed jar for a week or two without uh, going real funky. Um, to keep a healthy sample, I find it to um, try and get a lot of plant material and not a lot of junk from the bottom. And the plants, as long as they get sunlight, seem to do well, and the sample will just sit there and not kind of putrefy. I have had samples that have gone horrible almost overnight, and those have been all from Lake Ontario. Anytime I take water from Lake Ontario, it has like a 5 hour shelf life before it goes bad. Lake Ontario has a bit of a pollution problem. So it looks like the fly larva. Oh, there he is. I spoke too soon. So this is an insect larva of some kind. I won't be able to tell you what type of fly it is, um, but compared to most anything else, you'll see they're always enormous. They look kind of like a weird dragon. The head parts or mouth parts usually have large jaws on them. If he holds still for long enough, I might be able to take a look. But being an insect, it's actually quite mobile. Their metabolism and their size lets them move around quite a bit more than most of the other things you'll find. The microscope is an eBay special. It's listed as a pathological microscope. You'll also see it as a veterinary microscope. Piranha, yes, all those little roundworms were feeding on what I believe was a Daphnia. It was some kind of small freshwater crustacean. K&W, the problem with leaving samples around is if it's open to the air, they tend to evaporate, and if they're sealed off, they tend to run out of oxygen. Um, if you have enough plant matter, they'd probably hold up, but I'm not sure whether I could do one on a slide. It's something I'd like to see if it's possible. One of the tricks you can do is put a little Vaseline along the edge of the cover slip so that when you drop it on the slide, it makes a seal all the way around. It prevents the water from evaporating. Um, and I've had slides that have lasted for about 2 days, 3 days, um, but the bacteria growth goes crazy um, after that. You'll see lots of spiral bacteria, um, lots of just little dots all over the place in its bacterial growth. And eventually, all the larger animals either run out of food or oxygen. It's not really a great thing to look at, and it's not a great thing to have to dispose of, because that's a lot of bacteria that's been cultured in one place. And if it's been in an oxygen starved environment, it's something that's probably going to eat people from the inside. So it's not really ever a good idea to culture bacteria on purpose. I'm going to see if I can find those worms again that we're feeding. I thought that was very interesting. Um, so to align the microscope, uh, each each objective lens threads into a little turning piece at the bottom that lets you change between magnifications, and as they are, the center of each lens is not in the same point relative to its threads, just because it's such a tiny. We're looking at such a tiny picture on the with the microscope. So each lens has a number of adjustment screws that lets you move the actually lets you move the glass lens inside the housing. So the idea is you try and make it so as you change from one objective to the next, the object that's in the center of the frame stays in the center of the frame. Like as I change here, it's pretty close. But when I go up to the next magnification, I have to kind of pan around a little to find whatever I was looking at. Here you can get a real good look at the mouth parts on these roundworms as they eat what's left of this poor little crustacean. Oh, and one more thing about culturing or growing your own little protists. I believe there are methods to do it safely without having to worry about bacteria, because you can buy um, you can you can buy laboratory grade protists, particular ones that are used in biological sciences. So there are ways to do it. I'm just not sure what it is. Um, and if I were to try it, I'm definitely going to end up with something that looks more like what ends up in the back of the fridge or forgotten in the fridge, rather than a BBC documentary. All right, so to finish on less of a morbid scene, let's let's see if we can find those Stentor again, and then I'm gonna sign off. Oh, here's what I'm talking about. This is the edge of the slide, this line, and this bubble that's lit up with a little bit of color there, that's actually as the water evaporates inwards from the edge of the slide. You lose a little bit of area. So these slides kind of have a time limit from when you make them to when they become not particularly useful. Using the Vaseline along the edges of the cover slip does actually help quite a bit. Here we go. Here's a lot of rotifers all congregated in one spot. So I'm going to turn off my webcam. I'm going to let this run for a couple minutes before I sign out. Thanks again for watching. I'm glad you guys all enjoy this as much as I do. And hopefully I'll have some new material for you guys other than the microscope coming up. One of the things I've been working on is Schlieren photography or Schlieren video. So if you give me just a moment, I'll bring up my first little test run. I finally managed to find a telescope mirror for less than $200, and I was able to get Schlieren imaging to work fairly quickly. So this is my first try. I'm using a blue laser to illuminate the mirror. So this is the plume of just butane coming from a lighter. And there's the actual ignition. And the interesting thing, because this is a jet lighter, it actually makes a fairly laminar plume. It doesn't have a lot of turbulence in it. So moving the lighter down a little, you can see the turbulent mixing of the hot gases with the air in the room. The other thing it can do is see the warmed air around your hand. There's enough of a difference in the density just to see the presence of a warm body. So this should be sensitive enough to hopefully see things like supersonic shock waves from ping pong balls and whips, maybe. I haven't tried yet, but hopefully we'll be able to do that as well. Alright, that's it for now. I'll be in the live chat for a couple minutes, and then I need to shut it down. So thanks for coming by, thanks for watching. I'm glad you all like this as much as I do.